In this movie, I'm going to explain channel rhodopsins and how channel rhodopsins allow investigators to manipulate neuronal activity with light. This is a relatively recent development, within the past decade or so. It is very cool and very, very powerful. Neurons are normally excited or inhibited by the release of neurotransmitters from the cells that innervate them, or their activities can be manipulated experimentally by applying either depolarizing or hyperpolarizing currents directly to the cells, thereby exciting or inhibiting them. However, recent developments now allow microbial rhodopsins, the rhodopsin that algae and other microorganisms use for sensing light, to be inserted into neurons. These microbial rhodopsins, which are collectively known as channel rhodopsins, allow neurons to be excited or inhibited by light. All organisms sense light with rhodopsin. We will discuss rhodopsin in much more detail when we consider the operation of the retina. But for now, one of the microbial rhodopsins, called channel rhodopsin 2, or CHR2, is shown at the top of the slide. Channel rhodopsin 2 was discovered in a green algae, where it triggers photoorientation movements of the flagella that allow the algae to swim to lighted areas that are optimal for photosynthesis. It is called a channel rhodopsin because it contains rhodopsin, but it also has an ionotropic channel. As illustrated here, the channel is opened by blue light and is permeable to both sodium and calcium. Thus, when the channel is opened by light, it causes the influx of positive charges, resulting in excitation or depolarization. The other channel rhodopsin is called halorhodopsin. It occurs in a prokaryotic archaebacterium, Natromonius feronii, hence the name Natromonius feronius halorhodopsin, or NPHR for short. This organism lives in an extreme environment in salt lakes in Africa, where the salt concentration maintains its osmotic gradient by filling itself with chloride using the halorhodopsin, which is a light-activated chloride pump. As illustrated here, when activated by yellow light, the halorhodopsin pumps chloride into cells, thereby causing the cell to hyperpolarize. The halorhodopsin pump is really remarkable. In the presence of yellow light, it will pump chloride into cells even when the cell interior has a higher chloride concentration than the extracellular fluids, or when the interior of the cell is more negative than the chloride equilibrium potential. This amazing protein can drive chloride into the cell to concentrations that are 50 times higher than normally found in neurons. The genes for channel rhodopsin 2 and halorhodopsin have been cloned and can be transfected into mammalian cells, typically with a viral vector. As shown at the bottom left of this slide, the gene for channel rhodopsin 2, or halorhodopsin, is linked to a cell-specific promoter that is active only in certain types of cells, such as neurons. The construct is then engineered into a viral vector. The viral vector is then injected into a region of the brain, and the neurons in that region are transfected with the microbial rhodopsin. The transfected neurons then express the functional microbial rhodopsin, either the halorhodopsin or the channel rhodopsin 2. After the neurons in the brain have been transfected with the microbial rhodopsins, the neurons can be excited with blue light if the neurons contain channel rhodopsin 2, or inhibited by yellow light if they contain halorhodopsin. This slide shows a neuron transfected with both channel rhodopsin 2 and with halorhodopsin. The way the neurons are excited or inhibited with light is shown in the top portion of the slide on the left-hand side. Here, a rat has been implanted with an optical fiber through which laser light can be shown. The fiber is inserted into a light guide that is attached to the skull. The light guide can provide either blue or yellow light. If blue light is shown on the neurons, they are excited and fire action potentials. If yellow light is shown on the neurons, they are inhibited and are prevented from firing due to the influx of negatively charged chloride ions that hyperpolarize the cell. The stimulation of transfected neurons with light is called optogenetics. Using optogenetic techniques, experimenters are able to manipulate the activity of a particular brain region to evaluate its role in information processing, or even to evaluate how that region of the brain controls behavior, as we shall see in the next movie. There is a final point I would like to make about the transfections. Specifically, the investigators are actually able to see all of the cells that have been transfected. The way they do this requires some additional information. As we shall see when we consider the topic of the structure of the cortex, scientists have isolated and cloned the genes for a protein found in jellyfish that emits a green fluorescent light. That protein is called green fluorescent protein, or just GFP. The GFP is a fluorophore. What triggers the emission of the green fluorescent light is the absorption of blue light. The gene for GFP has been cloned, and indeed it can be attached to the microbial rhodopsin gene, which is then used to transfect a region of the brain in the same way as I described previously. So now every cell that's transfected with the microbial rhodopsin also expresses green fluorescent protein. In mammals, the brains of the transfected animals can be evaluated histologically after the experiment is completed. In other words, the animal is sacrificed, its brain is removed and sliced into thin sections. The sections can then be examined under a microscope, and the transfected cells glow green under blue light. For example, this slide shows a section from a hippocampus that was transfected with green fluorescent protein. Here, under higher magnification, one can see the cell bodies, the dendritic trees, and the axons. Notice that the entire cell expresses GFP, and the entire cell would also express the microbial rhodopsins, which include the axon and the axon terminal. Just as an aside, scientists have now modified the GFP gene so that the genetic variants can fluoresce yellow, or red, or any other number of different colors. The extensive expression of the microbial rhodopsin, however, means that investigators can excite or inhibit the cell bodies of neurons, or alternatively, they can excite only the axon terminals to assess the role of a particular group of transfected cells on their targets in a highly restricted region of the brain. You will see the significance of this feature in the movie titled Engineering a Memory with Long-Term Potentiation and Long-Term Depression.